Okay, all right, let's take a look. Now, we mentioned before in terms of epigenetics, one of the things that can be done is the methylation of histones. Methylation, of DNA. Mostly it's cytosine residues. There are these enzymes called DNA methyltransferases that put the methyl groups on. There are DNA methylases that remove them. For the most part, these are going to be long-term and heritable changes. DNA methylation generally inhibits transcription. In fact, it pretty much shuts the thing completely off. So that's one kind of thing. DNA methylation was the first epigenetic mechanism discovered. And once again, the way this thing can happen, the way it gets transmitted, is here you have, I'm just doing one strand here, M for methyl group. What's going to happen is the methyltransferases bind very strongly to the methylated, bind very strongly to methylated bases, and then they reach across the replication fork and actually methylate this sister as we're forming this new strand here. So we have another one that can do that, put one over here. And likewise, we have the same thing done over with this strand here, this new strand here. The methyltransferase reaches over and methylates that. So that's how we transfer this information from one generation to the next. The methyltransferases bind the methylated DNA, reach over, and the newly synthesizing strand on the other side gets methylated exactly the same place. And of course, this has to happen during the process of replication because the strands are separated, but they're still physically close together, so the proteins can actually reach that distance right at the replication fork. Okay, so that's what we saw here. So this is one way of heritable changes of this type of stuff. Okay, now, the second thing that happens in epigenetics is now we're finding more and more that the histones, recall that histones are the proteins that are the major structure of chromatin. The DNA wraps around that histone core, H2A, H2B, H3, and H4 to form what we call a nucleosome. The nucleosomes, with the assistance of histone H1 and its close variant H5, are going to assemble together to form that 10 nanometer fiber and then twist around or zigzag or whatever to make a 30 nanometer fiber and so on. So much of the structure of chromatin is dependent on these histones. We also mentioned before that histones are inhibitors of transcription. And the reason why is they bind DNA so tightly that other things like transcription factors and stuff like that simply can't get to DNA sequences that are wrapped around a nucleosome. So for transcription to take place, we have to do things like phosphorylate and acetylate the histones, and then chromatin remodeling complexes are going to be able to push them out of the way, push them up or down the DNA strand, and now we can get our transcription factors and what have you in the basal complex that bind. So in general, histones inhibit transcription, but they can be modified many, many different ways. We already talked about things like methylation, or rather, we already talked about things like phosphorylation and acetylation of histones. In general, what that does, it reduces their binding to DNA because, of course, histones are very highly positively charged. And that means you're going to be able to push them away, things like that. So generally, you have to do a lot of that before transcription takes place. But there are other kinds of modifications of histones as well, especially in what's called the tail regions of the histone. Because in general, a histone has sort of like a globular-like structure at the core, and then these extended sections, the tails that stretch out of the nucleosome and stick outside. So if you have a nucleosome here, the DNA is wrapped around it, and you got these histone tails sticking out of it in various places. These tails can be modified in numerous different ways, and the laundry list of things that we can do here is going to be, well, of course, phosphorylation, acetylation, And phosphorylation, acetylation generally increases the rate of transcription, but we also have other kinds of things that could be done. For instance, we can methylate histones. In other words, add these methyl, these hydrophobic groups. And they go on the amino acids lysine or arginine. Now when you do that, when you methylate the histone, depending on exactly where you stick the methyl groups on, what's going to happen here is going to be, you may increase transcription or you may decrease. It just depends exactly where you stick the methyl groups on. So it can have either effect depending on the exact location of that. Now, on the other hand, we have a couple other things. We have what's called ubiquitination. Now, that means covalently attaching that little small protein called ubiquitin to histones. Those of you who remember cell biology, when you want to get rid of a protein, you covalently attach a chain of this small protein, ubiquitin. And then that was a tag for it to get shipped to proteasomes and ultimately destroyed. But ubiquitin is not just used as a death sentence for a protein. If you add like one ubiquitin to a certain place, it may have a different effect. So when we're adding ubiquitin, that's going to be on lysines, that's another amino acid. When you add ubiquitin to it, we're not tagging this histone for destruction. We're actually regulating it, and in general what's going to happen, we don't know why it works, but in general, and here again, it can increase or decrease transcription depending where we're doing it at. And another modification, something called sumylation. Now, sumo, it's not a Japanese, a big overweight Japanese wrestlers. I don't know what it stands for, but sumo is a close relative of ubiquitin. It's a small protein that can get covalently attached to other proteins, and its main function is regulation rather than tagging things for destruction. It's very similar to ubiquitin in many regards, but it has more of a regulatory role. And once again, we stick these guys on lysine, and once again, this can have variable effects.
L-Y-G
one sec. Okay, L-Y-A-T-I-O-N. I told you, I taught handwriting in school all the time. I did that routinely.
Dr. Sauter,
Yes.
so all of these are able to untouch histones, all of these,
What was that?
like they're able to, I guess, somehow unattach histones, or exactly what are these doing?
Well, enzymes are going to attach these different things to specific sites on specific histones. In other words, in the nucleosomes, the histones that make up the nucleosomes, you can modify them. And generally you're going to do it at that tail region, right over here. And these are the parts that actually stick out of the nucleosome core. And then that has various kinds of effects. Now, exactly how this stuff works, we're still not really sure about it. It's relatively recently discovered.
Is like that sumo word, is
Yeah.
that like a form ubiquitin or?
It's a relative of ubiquitin. It's quite similar in sequence and stuff, but it's not the same thing. As far as I know, sumylation has been this relatively recent thing that's used to regulate certain types of proteins. As far as I know, it's not a tag for destruction like ubiquitin is. But what we've seen here is ubiquitin can also be used for regulation. So
So ubiquitin in this case, it's not,
Yeah.
doesn't tag.
Yeah,
destruction.
it's not, yes, in this case, when you want to tag a protein for destruction, you put a chain of several ubiquitins on. Here we're just sticking one on a specific amino acid rather than putting a whole chain. Put a whole chain on. If you wake up on a week or Thursday and have a chain of ubiquitins, okay, you can have a really, really bad day. If you have just one, it's going to ramp up your mental capacity and you'll get an 80+ on this. How about that?
Well, that's,
Okay.
So these can attach actually to the histones to either increase or decrease?
Yes, yes. Now, how they work on that is something we still don't know. But they see histones can have these different modifications, and they can be done and they can be reversed. But, and we see that, oh, if histones are heavily ubiquitinated or something, in one case we see transcription go up, in another case we see it go down. We don't know how that works yet. This is relatively new stuff. It's only been the past couple of years where we've been starting to see like reviews and stuff about histones and epigenetics. So this is a hot field. And there's a lot we don't know about it yet. But anyway, so we see all these different kinds of modifications. They can be done and they can be reversed as well. But these types of things too can also be passed down from one generation to the next. Especially stuff like methylation and ubiquitination, stuff like that. So these they're not necessarily, but they can be heritable changes. They can pass to the next generation. Now, how are we going to do how are we going to inherit modifications to the histones when we start dividing the DNA? Well, let's kind of do it like this. Now, without doing all this stuff, without doing all the arachnid stuff around the nucleosome and stuff, what I'm going to do is I'm going to draw these balls. These balls are going to have, okay, I'm kind of coloring these balls in a little, okay? Now, these are the original nucleosomes. Now, when the DNA is replicated, you get this replication fork again. And now we're starting to make our second strands here, okay? All right. Now, what's going to happen is half of the histones will go here, and half of the pre-existing histones go there. So the histones on the original DNA, or rather, the nucleosomes on the original DNA strand are split and passed. Half of them is passed here, half of them is passed here. So what we see now is an old original nucleosome here, another one here, and another one here. Now, on the other hand, there's still these kind of empty spaces here that we need to make. This is where we put the new nucleosomes in. And I'm going to represent the new ones by open circles. So this is old, or we'll call it parental. And this is new. Now let's suppose these nucleosomes have a bunch of modifications. I'm going to put M here to represent that they're methylated. It could be any of the other things too, by the way. Now, here again, what's going to happen is these modifying enzymes, say a histone methyltransferase, an enzyme that methylates particular regions of a histone in a nucleosome. Here again, or the ubiquitin ligases that add ubiquitin things. These enzymes recognize and bind to the already altered nucleosomes. So a methyltransferase will bind tightly to a methylated histone, and it won't bind very well to an unmethylated one. So, binds here, and then what it will do is then, the active site's over here, and now it methylates this new histone. So here again, we have a couple methyltransferases over here. They reach out and methylate these histones. So in this way, in effect, you use the methylation or the modification pattern of pre-existing histones. They're spread and distribute it to both sides of the replication fork. You use those in effect as a guide telling these enzymes, okay, these things are methylated here, the new ones methylate them in exactly the same place, or whatever modification you choose to do. So once again, these patterns can be passed from one generation to the next. Now that is not necessarily the case though. Sometimes these are going to be passed to the next generation, sometimes they're not. Now all these different changes in the histones are generally fairly long-term. They are reversible. So we can have enzymes, if we decide we don't want methylated histones, we have enzymes that can get rid of these guys, or whatever other kinds of alterations we're dealing with. So they can be stable, they can be very long-term, but if we want to get rid of them, we can alter them. Now, that brings up an interesting thing here. Cloning. Okay, at one time they said it couldn't be done. Back when I was taking development, up until the early 1990s, the best thing in cloning a vertebrate organism, somebody took some real early embryonic gut cells, just shortly after gastrulation from a frog embryo, took the nucleus out of those cells, stuck them into into a frog oocyte, and managed once in a while to get a clone. If they took anything that was from a later embryo, much less from an adult, forget it, just didn't work. And then that little trick to make Dolly the sheep. Okay. All right, well, nowadays we've cloned numerous different mammals. People have cloned sheep, pigs, cattle, rats, mice, dogs, cats. In fact, there's a company, if you have lots of money and really like your dog, there's this company that for a mere $250,000 would clone Fluffy for you. No, I'm serious, because I think it's based in Texas. And they do have customers. Now the problem with cloning though, and nobody's been able to clone a primate so far. The problem with cloning though, is if you're lucky, 1 out of 100 clones will actually make it to birth. And many times, what's going to happen is even after birth, they have defects and things like that, developmental abnormalities, sickness, premature aging, premature organ damage and stuff like that. Like Dolly the sheep, she tend to live about 20 years or so. That's in the wild, that's normal. Now on the other hand, you take Dolly, they put her down after about 6 or 7 years because she was getting very sickly, showing a lot of signs of premature aging and stuff. And that's typical of a lot of cloned animals. So your $250,000 clone dog might not live more than 4 or 5 years or something. Now what's going on here? Why can't you effectively take that nucleus and get it to express all the genes you need to express in a developing embryo? One of the big problems, there's two probable issues. One of them is if you're taking a nucleus from a somatic cell, that nucleus, it's probably its ancestor divided a long time ago. So it's been through multiple rounds of division. You're getting a DNA damage and stuff like that. Mistakes in cell division, mutations, getting zapped by cosmic rays from space, chemicals in the environment, what have you. So you got damaged DNA a lot of times, just from environmental stresses. Now, a mature differentiated cell, the 20,000 genes or so you got, maybe it's only going to express 5 or 10,000 of those. So if you got a gene trashed that you're never going to express in those cells to begin with, you don't have to worry about it. It's not going to do you any harm. It's just something that you're never going to express anymore. On the other hand, if every single gene is called on in a descendant of that cloned egg that you've done, and you're missing genes, that's going to cause problems. So that's one thing. Cumulative DNA damage that you're going to find in an adult cell that you're not going to find in oocyte. But the second thing is this. There's these epigenetic patterns, DNA methylation, histone modifications, and stuff like that. They are hard to reverse. Now the environment, the proteins in the cytoplasm of an egg cell, of an oocyte, seem to be able to do at least a partial reverse, but you have to reverse all this stuff because you don't want some gene that in one cell type is absolutely essential to make a normal differentiated cell. You don't want it strongly repressed because of DNA methylation, histone modification, what have you. So you have problems with gene expression in different cell types as this embryo develops. Many times they're lethal. Like I say, in typical clones of mammals, if you get 1 out of 100 live births, you're doing lucky. To get Dolly, it took 400 attempts to get a live birth. The rest of them spontaneously aborted in everything from early embryonic life to fairly far along in mother's pregnancy. So that's the kind of thing, and a lot of it is probably trying to undo all these epigenetic modifications. Okay, so that's a big issue. And it may even be more dramatic for primates, because although people try to clone monkeys, Congress needs more people in there. Okay, people have tried to clone monkeys, but nobody has succeeded so far. Even though they've succeeded in a lot of other mammalian species, nobody's pulled it off yet. Okay, well, so these things can, but not necessarily are, heritable. And now we ask the question, why do we do this kind of stuff? What's the function, especially heritable or long-term changes? Well, there may be a pretty significant evolutionary function for this type of thing. That is sort of responses to the environment that you can pass down to the next generation, or you can respond in utero. In other words, while you are developing, you may be able to make changes that will allow you to face a probable environment in the future when you're born. Now, case in point, let's suppose this, and there's some evidence, it's been shown, I think, in some animals, and there's some evidence that in a few incidents in humans, this may have been the case. Over a human incident. After, let's see, actually 2 of them. One of them, after World War II in Europe was devastated. The Nazis, came in, trashed everything. The Allies bombed every city to the ground. The Russians trashed everything. Europe was in shambles. The whole country, the economy had collapsed. The infrastructure gone and stuff like that. So in those first few years after World War II, things were very tough in Europe because the nations were all devastated by World War II. The cities gone, the infrastructure gone, what have you. Not surprisingly, food was very, very scarce in those times. It was hard to get by. One thing that did help is the Allies, the United States especially, put in a great deal of effort, the Marshall Plan as it was called, to try to support and get Europe back on its feet again because, well, it wasn't just being nice. The main thing was they were afraid that all this kind of deprivation stuff would give communists a huge advantage, and we would have the Soviet states of Europe in a few years if we didn't do something about that. So that was one of the big things. And they spent the equivalent of probably trillions of dollars in current terms to get Europe back on its feet. Okay, well anyway, during that brief period where hunger and famine was common, and in Sweden around 1800 or so, there was a mass famine that caused thousands of people to starve to death. And here again, during that brief period, it was seen that people that were fetuses during that period, in other words, they were born shortly after that period, tended to have an unusually high rate of obesity. In other words, they got fat really easily and really quickly. And people that were born before that or after, or significantly after that, didn't have as much of a problem. So the question, what was going on? Well, one thing after a short while, Sweden, of course, became, after that famine, they became in pretty good shape. Europe, after a few years in the devastation of World War II, they started building up their economy, and within a matter of a decade or so, they became wealthy and prosperous nations like they are today. But that brief cohort of people in both these instances seem to have an unusual problem with obesity. Now what happens, of course, if your country is rich, it means food's widely available, cheap, and you can eat whatever you want, and however much you want. What might have happened in this, there's no proof of this, but there does seem to be some interesting connections. What might have happened in this situation like this, and we have seen some things in animal studies. Okay, here you are, you're a fetus. Mommy's hungry. You know that because you're not getting the nutrient supply that you would ordinarily expect. So in effect, what mommy's kind of telling you without even realizing that is that life is tough outside. Food is scarce. So what you might want to do is, if you can, is alter the expression of certain genes. Genes that may control appetite, that may control energy storage in the form of putting into fat and stuff like that. Those kinds of things would help you survive in a time of famine. Whatever it takes. Stingy metabolism, high appetite, when food's there, eat it, and like there's no tomorrow because there probably won't be. In other words, in effect, in utero, the fetus is genetically preparing itself to meet the challenges of the environment when it's finally born. Now, okay, so it's got all these nice stingy genes and high appetite and all this kind of stuff, and now all of a sudden you get into an environment where food is plentiful, food is cheap, you can eat whatever and how much you want, and lo and behold, you're going to overeat. You're going to put on the pounds, big time, compared to people that did not have those changes. Now, they've done these different kinds of things where they expose, in mammals, expose them to certain types of environmental stress. And they do find, in many cases, that the newborns and their, that the progeny have different changes. And apparently, I guess in some cases, different patterns of DNA methylation, stuff like that, different changes that allow them to face the challenges of the environment. So that's one of the possible roles of epigenetics, is allow the embryo to sense what the probable environment is like, and before you're born, before you hatch, whatever the case is, you make the appropriate changes in gene regulation to face those challenges. Now, there's a lot of stuff we don't know about that, but that could be a big thing about why we do this type of epigenetics. That it allows you to prepare yourself for the probable environment you're going to end up in when you're born or hatched. So that's another kind. And here again, if you have other kinds of environmental changes and stuff that may be long-lasting, you may make epigenetic changes later on that will allow you to face the challenges of the environment. So that's an interesting thing. Okay, now case in point, just what this could imply. Let's suppose we have this kind of thing where, you make changes to deal with food scarcity because you've sensed in utero or in the egg or whatever that life's tough out there. Now, if you're born, you go into that food scarce, that environment of famine, you're going to have a head fight because you've changed the expression of genes controlling metabolism, appetite, fat storage, that type of stuff. You're going to be in better shape than a competing animal, competing organism, or your neighbors or whatever that have not made those changes. They're going to be more prone to starvation than you are. So that's a benefit. That means you're more likely to be able to reproduce and spread those genetic changes and those ability to make these kind of genetic changes into the next generation. So there's advantage to it. Now in cases where things rapidly change, like post-World War II Europe to, modern Europe and stuff, that can actually do more harm than good. But, who could tell at that point in utero, hey, mommy's starving, mommy's really hungry, I better get myself ready for this, and then all of a sudden there's food up the wazoo, and now, you're huge. Okay, so that's the kind of thing, and it might, one of the functions may be to allow you to sense the probable environment and make the necessary changes to be able to meet those environmental challenges by regulating, ramping up or ramping down the expression of certain genes.